All right, so good morning, everyone, and welcome to the CAE-CET weekly seminar. It's my really great pleasure to introduce you today to Vivek Prakash, Dr. Vivek Prakash, who is a professor in the Department of Physics here at the University of Miami. He also has secondary appointments in biology and marine biology in Erasmus. I believe he can tell us more about that and how he got those. So he got his bachelor's and master's in engineering mechanics and mechanical engineering in India. So he's actually an engineer deep inside. He got his PhD in applied physics from the University of Twente in the Netherlands, and he did a postdoc in Stanford. And as a good physicist, he basically switched fields. Now he does curiosity-driven research at the intersection between physics and biology. And today he's going to tell us more about the biomechanics of simple marine animals. So Vivek, thanks for coming, and the floor is yours.
Great, thanks so much, Lewis. Thanks for this kind introduction. I'm really happy to be here, and I'm really excited to have an engineering audience look at this talk. It's been a while since I did that. So hopefully slides look okay, but otherwise please stop me. I would love questions, so feel free to interrupt anytime. Definitely, that's totally fine. And I'll try to go slowly and kind of introduce these animals quite a bit, because if you're from engineering context, you probably have never seen these animals, right? So I want to convince you that simple marine animals can be fascinating, and you can really use mechanics as a tool to really understand the life cycle of these animals, how they actually swim, feed, etc. Right? So I want to show you that they're fascinating. So I'm in the Department of Physics and have secondary appointments in biology and marine biology. So you can see I have a very interdisciplinary approach to how I approach science, basically. So let me start with the big picture of the Earth. So you guys know that the Earth is actually covered more with water than with land. More than 70% is actually water. And life actually began in the oceans, right? So here's this beautiful mandala of plankton. So if you look at this, it's really amazing, right? So this is all the planktonic animals, which are like freely floating animals in the ocean. That was collected by this Tara Oceans expedition, which went all around the world for about 2 years, collecting different types of plankton. So there are these really diverse, enigmatic, exotic organisms which hold many, many secrets waiting to be unraveled. So this really drives me, my research. It's really exciting. So now, when you have these organisms, right? So there's so many variety of them, you can classify it in different ways. So one way is to look at their length scale. So going from the smallest scale to the largest scales, you first, small scales, you come across nucleic acids. Then at the micro scales, you come across single-celled organisms. Now, when you go to higher scales, like larger length scales, at the millimeter scale, you come across, for the first time, multicellular animals and embryos. And so this includes, and you keep going up in scale, you go all the way to humans, and of course, the big blue whales, right? So my interest is in this range of 100 microns to about 100 millimeters. And this is primarily where you have multicellular animals and embryos coming in. Now, another way to classify animals is to look at early branches in the animal tree of life. Now, again, think of it as an animal tree of life, and our first unicellular relatives gave rise to the first animal phyla. So these are the ancestral animal phyla. And in this diagram, as you go towards the top, you increase in complexity, right? So most of you have heard of jellyfish. So they're cnidarians, and bilaterians include everything else, including us, right? Now, these three are the basal ancestral phyla of animals. This include ctenophores. So like comb jellies, different than jellyfish, but kind of similar looking. These are sponges, and these are placosomes. And my focus is on these simple marine non-model systems. And so I'm going to tell you more about placosomes, and ctenophores are something that is kind of my long-term interest. So the question is, why should you be studying these things, right? So why should you study simple marine animals? the key is, the reason is that there are new questions to ask and new opportunities. So it's very exciting. They are clearly understudied, and there are so many secrets waiting to be unraveled. You have the ability to bridge scales. You have the ability to go all the way from cells to tissues to entire organisms, and you get to skip the whole organ system part because these animals are so simple. You get to ask questions on the origins of multicellularity and origins of complexity, which are really exciting, right? And now you also have new approaches and new tools available. For example, there are so many advances in molecular biology and genetics, and you can carry out in toto imaging. That means you can image all the cells in entire organisms at once. And there's a huge interest from the physics community, soft matter communities to study emergent mechanics in these types of animal systems. So in one sense, we look at organismal scale biophysics. And the big, of course, question is, how does physics shape biology? So that's the broad question. And as you can imagine, it's such a broad question, we have to start focusing on something, right? So my focus is on flows in living animal systems. And the reason why I am interested in flows is, as Louis said, I started as an engineer. So I did my undergrad in mechanical engineering, and I got interested in fluid mechanics ever since my junior year. So I started doing research, and then I shifted my focus in graduate school to fluid mechanics. So I did my PhD in applied physics. My thesis was very much classic fluid mechanics project. I looked at light particles or buoyant particles in turbulent flow. So I did my PhD in the Netherlands, University of Twente. So I looked at how air bubbles behave in a turbulent flow. Okay, so this is air bubbles in water, light rigid spheres. And for my experiments, I used a large scale setup, and setup looks like this. So this is a 3-story facility. That's the first floor. It's a water tunnel. We're injecting bubbles there. And now we're lugging this camera to the top. This is the second floor. So that's me. 10 years ago, I had so much more hair. And this is now the third floor. And as you can see, this is a closed loop water tunnel facility. And it's entirely designed. It's a very unique facility designed to study turbulent flows under very well-controlled conditions. So this is my entire grad school in one slide. So what I did was come up with or develop quantitative experimental techniques to track thousands of particles, both in 2 dimensions and 3 dimensions. So I did both 2-dimensional tracking and 3-dimensional tracking of different systems. And I asked classic fluid mechanics questions on what is the effect of density, what is the effect of finite size, gravity, void fraction, which is how much gas volume fraction is present, and do the bubbles cluster, right? So this is all really interesting, very new stuff, pretty exciting, but I was looking for more adventure, so to speak. So I used to come to these conferences in the US, the APS Division of Fluid Dynamics conferences. So I was approaching my final stages of PhD, and then I used to go to all other talks, like all kinds of talks possible. And then I came across this talk where I saw this video of a jellyfish swimming. So this is from John Daveri's lab at Caltech, and they injected a dye, and the jellyfish swims upstream. And you can see all these beautiful fluid flow structures, right? So this really got me fascinated. So then I really wanted to be studying fluid flows around animals in my postdoc. So I wanted to switch fields. And another set of problems that I came across was, how do cells actually flow in tissues? So this is a video on the left of Drosophila development, and you're seeing single cells, they're actually sliding past each other, okay? And this is during development. So on the right side, you have a fluid mechanics problem, and on the left, you have sort of continuum or tissue mechanics problem, sort of solid mechanics problem, right? So I wanted to build on my PhD in fluids to really do biophysics. So this is when I transitioned into my postdoc at Stanford University. And these 2 themes still continue to be my areas of research interest, which is flows of cells in tissues and fluid flows around animals. So there are 2 recurring themes in my research. So one is to connect form versus function. So the idea is to connect the form of an animal, which could be its geometry or morphology, to its functional behavior. And function could be feeding, motility, reproduction, or development. So my approach is to use mechanics as a tool to relate these two things, to relate form versus function. So we're very much interested in the biological question, but we really want to use mechanics as a tool. And another emerging important theme is the idea of emergent mechanics. So how do phenomena at the smallest scales give rise to phenomena at the larger scales? Right? This is a very commonly used, commonly studied theme in physics, right? And in biology. So what I'm going to do today is, this is the outline. So I'm going to tell you two stories. Both are from my postdoc. I kind of started new here at UM 2 years ago, and my lab is still being constructed. So maybe those stories will come in a few years' time. So what we're working on currently. But these are stories from my postdoc research. So first I'll tell you about the tissue mechanics problem. So this was again my postdoc lab. I worked with Vivek Prakash. And if some of you are wondering, we have the same surname. We are not related. It's just a common Indian surname. And this work is in collaboration with Matt Bull, a very, very talented grad student. So here, we are interested in understanding the extreme limits of tissue mechanics in living animals. And just to kind of get you started on the project, to give you a background. So you know that animals are crawling, walking, running, swimming. So the animal tissues are always under constant dynamic loading. So the question is, what are the limits of tissue mechanics in these time-varying force landscapes? And during development, so all of you know that development happens at a single cell level, then there's an embryo which forms. So there are these large-scale morphogenetic movements where there's large-scale tissue shape change. Usually, after embryogenesis, when an animal reaches an adult form, the shape is fixed, right? That's usual. But the question is, are there examples of adult tissues changing shape? So now you find a lot of weird examples and fascinating examples when you go to these really lower, simple marine animals. So here are some examples of animal tissues in dynamic force landscapes. So in jellyfish, people have shown that if you chop off one of its arms, the animal regains symmetry in just about 2 days. So it's an extremely fast process of tissue repair or symmetrization. And in Hydra, it's been shown that the mouth actually ruptures open every time the animal feeds. So that's another fascinating example. And there's also always, there is this medical relevance to this. So there are these diseases called acute respiratory distress syndrome, ARDS, which are actually caused due to a plasma membrane induced stress failure in the epithelium. So imagine while I'm right now talking to you, breathing, right? So there's alternate compression and expansion in my lung epithelium. But sometimes if there is a fracture in the epithelium, you get all kinds of diseases. So this is an active area of study. And the way they study this is using cell culture assays right now, because it's really hard to image directly into the human lung, right? So there's definitely medical relevance, but also fascinating marine examples. So we were interested in understanding the extreme limits of tissue mechanics in the living animal system. And for this, we chose animal system, a simple animal with a flat epithelial architecture. So this is the Trichophylax adherens. So as you can see, that's the video on the left, you're seeing the entire animal. You're seeing the animal from the top view, and the animal is crawling on a substrate. So this provides us two key advantages for this type of a question. First is you can do cellular scale in toto live imaging. That means you can image the entire animal and look at individual cells. So what you're seeing there is this firework pattern. So those are actually the fastest cellular contractions in the animal kingdom. And that my colleague Shahaf Armon showed that in 2018. So now it's also moving around. So you're seeing time-varying force landscapes due to its motility. What I did as a postdoc was to look at a larger animal, like the one on the right, and you see it's changing shape extremely, right? So it's changing shape. It has extreme shape change, and it's exploring its environment. This is like a 2 millimeter size animal, and it's trying to look for food. And if you use your imagination, you can start seeing all kinds of familiar shapes. For example, I see like a Snoopy shape here, right? So we know in our cultural dishes that these animals don't have a fixed shape like you would expect in most animals. So these are snapshots of animals from our culture dishes, and you can see shapes going all the way from a circular shape to this really long, elongated thread-like shapes. So the question is, how do these epithelial sheets undergo such extreme shape changes? When I started this project, we also knew there was one more interesting phenomena, which is how these animals reproduce. So these animals reproduce by asexual fission. So this animal in the center forms two regions, which basically start walking away from each other, right? They form a small thread, and now the thread breaks. So the special thing about this is a large animal. This is 3 millimeters. So there are millions of cells doing this, right? And how do all these cells collectively decide to come together to have this kind of a ductile deformation, right? So this is like forming a thin wire, right? And the other important thing to notice is the time scale. So this is an extremely fast process. It's like it took place in 1 hour. So that's faster than any known biological process. So to begin with, we kind of hypothesized that mechanics must be playing a dominant role in this, right? And we assume that there are locally cells rearranging themselves to enable this such fast extreme shape change. So here we are, this is what we knew, and we had to do this project. I wanted to understand cellular scale detail. Before I go into the project, I wanted to show you some more things about the animal, right? So this is a cartoon of the animal, and you can see it's extremely thin, but it can be several millimeters in size. So they're like thin walking pancakes walking on substrates. So this is a cut section. So it has a 3-layered tissue, right? The top and the bottom tissue layers have cells of different architectures. So in one sense, the mechanical properties would be different. These bottom hair-like projections are actually cilia, and the cilia are sticky. So this animal basically walks using millions of legs, right? So you can imagine what a mess it would be. Now, these animals are special. They're the simplest animals, the 6 cell types. They don't have neurons, they don't have muscles, no ECM, which is extracellular matrix, no basement membrane, no tight junctions. However, they do have adherent junctions holding cells together. So in both from a physics and biology point of view, from a fundamental point of view, it's a beautiful system. It's a simple, minimal epithelial tissue to help us understand what such a tissue system is capable of from a mechanics point of view. Okay, so this was about the animal. So now the question was, how do I do this? So for a model system like zebrafish or Drosophila, you're able to now image all the cells using fancy transgenics, and they have light sheet microscopes. But this is an understudied model animal system, right? So how do I get cell level resolution at such a large scale, right? That's an open challenge, because if you zoom in, you're only going to get a very small field of view, a few hundred microns, right? So we had to develop a new technique, and I came up with this idea. The idea is to tag the surface of the animals with sticky beads. So I started with fluorescent beads, coated a specific protein, a sticky protein. This is like a lectin, it's wheat germ agglutinin, and this specifically binds cell membrane. Okay, so I spent about half an year doing this. And now, so basically this is like creating sticky particles, sticky beads, and dropping them, sprinkling them like shampoo on the animals, and then washing away the supernatant. So the beads permanently stick, and now you can see, you can really tag the inside tissue of the animal, which enables you to carry out high-speed, long-duration particle tracking of thousands of beads. So it's a very powerful technique. We've also applied it in chicken embryos. Can be used in many, many different systems. So now that you have sticky fluorescent beads, you can do, this is what the raw data looks like. You can do large field of view, high-speed, long-duration tracking. So you can see inside the animal's morphogenetic flow field, right? Now with this, you can imagine all of our engineering background can come in, right? So we can visualize the trajectories inside, right, inside the tissue. So this is flow trace. It's a path line visualization technique. We developed that. You can go download an ImageJ version and get a video like this in like less than 5 minutes. Okay, so I encourage you to go and see that. Flow trace was visualization, but now we can do, of course, carry out a particle image velocimetry analysis. So this is quantifying velocity flow fields inside the tissue. So each of the arrows on the left is pointing towards the velocity vector. And once we have the velocity information, we can calculate the strain field inside, right? So you can see the strain field is spatiotemporally fluctuating continuously. So in other words, the animal is doing yoga, right? It's stretching itself continuously, and you have all these strains inside it. So in one sense, you can see that now we're able to quantitatively show that motility of the animal induces mechanical forces at the organism scale. So we call this like a self-inflicted rheology. So this was really nice. This was really cool. But then the question was, okay, now what, right? So we saw several data sets like this over many, many hours. And so this animal in the next 1 hour or so actually showed us something very unexpected. So this was a discovery. So this animal in the next 1 hour had a tissue fracture hole induced inside its bulk. So this is now a shear force, right? And I've highlighted this exact hole on the right side. So you see actually a hole opening up in the bulk of the tissue. So imagine, just think for a moment, because any fracture in an epithelium, like if I had a tear here, that would be a bad thing, right? Things would ooze out, and in general, fracture is a bad thing. But in this case, this is the first example of a self-induced tissue fracture. We're not doing anything to the animals, we're just watching them. So it's all physiological conditions. So now, after we saw like a shear force opening up a tissue fracture in its bulk, we were looking for other such examples. So we also found that a tensile force can give rise to a tissue fracture hole. So you see again these two holes opening up in the bulk of the animal. And if I plot the area of the hole, you can see that in about 2 minutes, it actually reaches the maximum size. So now this is like any of your material science problems, right? It's like an engineering material science problem. So this slide is actually not for you guys, it's for biologists, but you guys are better experts than me, but I'm just going to quickly go over this to say that like you test all materials and do carry out these stress strain curves, you reach first the elastic limit and then the breaking point. And this is all plastic deformation, right? Now, fracture mechanics tells you that there are principally 3 modes of failure. One is tension, the other one is shear, and the third one is out of plane shear. In our case, we don't have any out of plane shear. This is like a 2-dimensional tissue, but we do actually have mode 1 tension and mode 2 shear. So now we can start thinking of fracture mechanics in living animals for the first time. So this is the first example of such a thing. Okay, so now we have fracture mechanics. So these are large scale animals like 3 millimeters. So what we wanted to do next was look at what happens at the individual cell level, right? So we wanted to zoom in, and we did that. We went to confocal microscopy. So now we're looking at the tissue at a much zoomed in resolution. So now we're looking at the animal from below. Okay, so we're looking at the bottom epithelium, and we use 2 stains to actually tag the entire epithelium, like a green and a red. That's a membrane and a lysotracker. And what you're seeing is live, like this are videos shown at the bottom and then snapshots at the top. And these are time points. Okay, so here you see these small dark gaps. So these were actually micro fractures in the tissue, which existed at a time 0. But in about 13 minutes, they actually start growing in size. So they're coalescing, right? Now, this looks like a material science problem, right? Like if I didn't tell you it's an animal, this looks like any other hole coalescence problem. And in the next 10 minutes or so, those holes grow in size and this becomes a stable fracture hole. So this process just took 15 minutes. Now, the even more fascinating thing is in the next 15 minutes, these holes can actually heal themselves. So the edges, so this hole, the edges can come close and the hole can heal itself by kind of zipping itself. So this system is a really fascinating system to study tissue fractures and healing. And if you have any question here, it's probably going to be an open question. We know nothing about the system except for this happens, right? So it's very, very interesting and open-ended. So now that we did this, we of course are physicists, we're engineers. We want to make in silico model to see if you can understand this better, right? So what we did, and this is done by Matt, the grad student. So we made a simple model based on the experiments, and what we wanted to do was capture the ventral tissue, the mechanics of the ventral tissue. So we're kind of capturing this experimentally by making the sticky ball spring model. So here there are 2 cell types, the green and red, right? Ventral and lipophil cell. And the sticky balls are held together using springs. So the springs represent adhesion or adherence junctions. Now, if there's a spring, that means it's a normal bond. If the spring breaks, it's like a fracture, okay? And we're capturing all these aspects in the experiments, like biodisplicity, non-confluence, local cell deformation is neglected. Now, so this 2-dimensional sticky ball spring model, we're going to put a force and stretch it to see if we can understand our fracture phenomena better. And this is the model we started with. So this vertex model is like a standard model nowadays used in modeling all kinds of tissues. The one thing is we're going to focus. So this is all energy, and then these terms are we're going to focus on this adhesion term, which kind of models cell-cell contacts. Okay, and everything else is kind of known in literature. And we're looking at fast timescales. So this is something that people don't look at. They typically look at Drosophila. And in this adhesion term, we're trying to model the dynamics of cell-cell junctions. Okay, so that is our focus. And I can tell you more about these models if you want later. Now, what we did was come up with a phase diagram, and the two key parameters which are relevant to the system are how much force we put in, right? And the length at which springs break. So that's the Y and X axis. So in order to come up with this phase diagram, we did a lot of simulations. We looked at shear transformations, bond failures, and packing fraction. We put all of them together. Each dot is a simulation, and we kind of came up with this phase diagram. Okay. Now, if you zoom into this, we could actually identify 3 different material phases: elastic, ductile, and brittle. Okay, so depending on where you are in this phase diagram, you can get these 3 transitions: elastic, ductile, brittle. So now I'm going to show you videos from each case. So this is the elastic case. So you see we are pulling the tissue, but nothing much happens, right? You see these force chains which come up, the red force chains. If I show you the ductile case, you're seeing local cell remodeling, right? So the tissue is becoming thinner and thinner. Okay. And next, going to the brittle case, you see a tissue fracture hole coming up in the center, right? So now all of these were steady loading, right? Steady forcing. But in the real case, in the animal, we know that it's unsteady, right? So we did that. We did unsteady pulling. So you can get both fractures and you can get healing. So it's really amazing that this our simple in silico model can actually capture the essential phenomenon here, right? So that is what we did. And the next thing was, can we say anything about these material properties in this type of tissues? So we wanted to see if we can do further quantification. Remember, we're not doing anything from an engineering perspective. We're not stretching, we're not pulling, we're just watching the animals, right, at this point. So I did this non-affine motion analysis. So this is a metric that is used in soft matter physics in different systems like colloids. If you have a bunch of particles like this and you apply a small shear, at a later point of time, you can have ordered motion or slightly disordered motion. And this d-square minimum metric can pick up the amount of disorders, okay? So I did that, and you can see this metric really works well in this Recoplex system. And everything that you see in red color is like highly non-affine motion, and blue would be like uniform affine motion. So this nicely highlights shear bands in the tissue, okay? So how is this useful? So we looked at the correlation between high values of this d-square minimum versus the internal strain. So if you remember from the PIV stretching, we could calculate internal strain. So it turns out these high shear zones were correlated with the maximum internal strain, as you can see here, right? So we kind of use this to make this plot. But remember, we're not doing any engineering experiments, right? It's all indirect. So we wanted to plot the shear versus strain rate to see if this tissue is like a yield stress material, okay? And you guys know this. So yield stress material, a good example is toothpaste, right? If you when you press on the tube, that paste comes out, right? So that's a good yield stress material. So our experiment showed this trend actually, and we were also able to recover this in numerics. So it does indicate that this material is indeed like a yield stress material. And others have recently shown, like this is in zebrafish embryo development, they've shown that there is spatially graded yield stress. So this is a phenomenon that many people are finding in different systems. So this is like a new way of characterizing material properties without actually doing any perturbations, right? Just by watching and quantification. So now we did all this engineering stuff, and it's really nice, but then why do the animals actually use it? Why is it relevant for their biology, right? That is the question. So now we're going to look at longer timescale videos and see whether the animals actually is useful for the animals. So these are videos from our culture dishes, so taken over longer timescales. And you can see that there's a tissue hole in the center, right? These are native conditions in Petri dishes, and the hole already healed itself, right? So it's like a 1 hour process. So basically we could prove that fractures are actually physiological in these animals. So it's part of their life cycle. Okay. So what you saw was this time sequence. So in half an hour, there's a failure and then fracture, and then in about 1 hour, it heals. So if you look at the same animal after 5-6 hours, you don't see a trace of fracture, right? Now, one thing I should point out is all of these are ventral fractures in the bottom layer. Sometimes when a hole opens up in the ventral, the top dorsal can also have a fracture, and that looks like this. So the animal looks like a donut. Its hole is now expanding, and moving. And this is now longer time scales. This is like 3 hours, 4 hours. And it forms a small thread after a while, right? And the thread now breaks, as you can expect, right? So now you get this long, stringy animal, right? So this is what you just saw. The dorsal fracture forms, and then it becomes like this donut type animal. It forms a large hole, and then it becomes a stringy animal. This took like 10 hours, right? So with this mechanism of fractures, we're able to completely explain the shape space that we started out with, right? We can explain all the shapes that are possible. And clearly fractures are responsible for permanent shape change, right? So fractures are the fastest way to go topologically from this circular shape to this really long, elongated shape. And you have two types of fracture that is relevant to the animal. So fractures are physiological and useful for asexual reproduction in these animals. So here is a summary. We found ductile-to-brittle transitions in the tissues of the simple animal, which are all induced by its own motility. Fractures induce permanent shape changes, and we have a new technique to measure tissue properties in living animal systems. So this work was published recently. You can go look it up. At this point, are there any burning questions from somebody? Okay, so maybe we can talk about questions later. So just to summarize,
there's a question in the chat, Vivek, for you.
Yes, good. Oh, chat. So chat, when you miss tissues, is it possible that the light source induces fractures? Yes. Okay, so that's a good question. I did think of it, and in one of the experiments, in the first experiment I showed you, there was some slight fluorescence light source. But just to confirm that this is not an artifact, this is why we went and checked our native culture dishes, right? And then we started seeing so many fractures, and we had always noticed them before, but we never made the connection in our head. Like we always thought it's an accident or somebody poked it. But yeah, we confirmed that it's actually physiological phenomena. That's a good question. And these things, when you want to make a big claim like this, it's very important to back it up with good evidence. Yeah.
Do the fracture, like, does the fracture kinetics depend on the softness or the stiffness of the substrate they're sitting on?
Absolutely. All of these phenomena are highly dependent on how much traction force they can generate, right? So imagine when the animal is splitting or there's a fracture, two coherent regions of cilia need traction forces to pull each other apart. So if they're on a slippery substrate, they won't even be flat in the first place. They will be slipping all around, right?
So you're talking about slip, like friction. I'm talking about,
right.
Like, would they remain rounded up if it were on a very soft, like what kind of substrates do they live on in real life? Yeah,
in real life, it's like a solid substrate. It's in the ocean, you have, let's say, any solid substrate, and they live on a biofilm. And in the lab, they live in petri dishes where there's an algae biofilm. Having said that, they round up, they curve up, and they can even swim around. Not active swimming, but they can float around. And we've seen that they even can live upside down on interfaces. So there's a lot of things to explore there, like how they do that or why they transition.
And there are age-related changes to the stiffness or recovery mechanisms in these animals?
Yeah, so age, these are great questions, actually. So age-related meaning, so whatever phenomena we're seeing here is a very fast time scale. And we know that cell division time scale is like 10 hours. So that means there is growth, definitely, and we're not accounting for cell division here at all. Okay, so that is something we should do, and that's a more long-term study. How does shape change fracture mechanics come in with addition of new cells and also apoptosis, right? Cell death.
I mean, I'm just trying to find angles, right, where it could become relevant to aging-related questions in human health, or changes in substrate stiffness that also happens with age
Okay.
and human health.
Okay. Yeah.
Right. How could
Yeah.
this be a model system to answer NIH relevant questions? That's sort of where I was coming from.
Yeah.
Of course, it's curiosity based, but
yeah.
Okay.
Yeah, I guess my main from this animal, this system is really good for the fast time scale study. So definitely the how does epithelium have fractures induced in it from the point of view of ARDS type epithelium, lung epithelium. That is the best medical pitch I can think of right now. Aging wise, it becomes a more tricky thing because these animals, first of all, it's hard to follow them for a long time, like individual animal in detail.
But could you not get young animals and older animals?
Yeah, so age is a difficult thing. So in one sense, the age is technically infinite here because they're clonal animals, right? So the life cycle is they spawn as an animal, they grow in size, split. Again, small animals grow in size, split. So yeah. So technically defining age is also a tricky thing.
Very exotic.
Yeah, very exotic. And see, that brings up another important question, which is what is the development of these animals, right? So at some stage in evolution, a sperm and egg got fertilized, then they started growing. We can't capture development in the lab. Nobody can. We don't know what conditions they need. So right now, all we can do is we can only study these clonal animals. And if we can get them to develop, then we can do like genetic perturbations, transgenics, and so many other things. So that is not possible at this time.
Yeah. Okay.
Yeah. Thanks, Asha. Great. And there's one more question. Would water salinity change fracture dynamics? To an extent, yes. They're very sensitive to changes in salinity. Fracture dynamics in the sense, yes, you can vary the salt content, so the adhesion would be affected between the cells. And that is actually a nice experiment that we've done a little bit, but we should do that systematically. Yeah, good question. Good question, Shruti. Thank you. Okay, so I'm going to go on. So now, again, going back to the form versus function theme, right? So we saw that this flat form of flat tissue can utilize fracture mechanics for the function of permanent shape change and asexual reproduction. And again, the smaller scale fractures started coalescing, and it gave rise to these large scale fractures and like shape change, which is relevant for the animal, right? So now I'll switch to biological fluid mechanics. So this is a project on starfish larva. So this was again, I was helping William Gilpin. I was a postdoc helping William Gilpin, who's again super talented grad student. He's starting his own lab at UT Austin in this fall. So look out for things from his lab. He's fantastic. So here we're looking at development, and now just generally describing the development process from going from embryo to, let's say, a sessile adult. So let's talk of a marine system like a starfish, right, that you see here, right? So sometimes development is direct. That means the embryo can look like the starfish, but sometimes it's indirect. That means the larval form looks nothing like the adult, right? So this is sometimes a possibility. So we study this starfish. This is the Petiria miniata. This is a California native species. So that's what it looks like. And in about 3 weeks, they reach this larval form. And that in that larval form, it looks like a spaceship, right? It looks nothing like the adult, right? So now if you put them in a petri dish and you see from the top, this is what you see. So the larvae freely swim around for about 2 months before settling down, and they're less than a millimeter in size. So this is a sped up movie, and this is a low Reynolds number system, right? And you guys know Reynolds number, ratio of inertial force to viscous force. So if Reynolds number is less than 1, viscous forces dominate, right? So we did this experiment initially, which is to take one of these larvae, put them on a glass slide, put a cover slip, gently squish it, put some beads, tracer beads, and now do time-lapse imaging. This is what we saw. We saw this really beautiful array of vortices. These are like 8 vortices around this larva. And it was really fascinating. This really blew our minds, and we wanted to understand. It's like, oh my God. So this is completely unexpected. How do these form? Why do these form? That was William's whole question for next 2-3 years. And just to point out, in low Reynolds number systems, most of what we know in literature, these single cell ciliates or like this really small swimmers have like 2 vortices surrounding them in this frame. And here, now this is such complexity in such a small Reynolds number system. So it's kind of a unique system. So that's why I really got intrigued by this. So now I'll first talk about how these vortices form. So if you look at the geometry, the larva is like this. So these are two hoods, and the mouth is actually in the center. Okay, here. And what you see on the periphery are these bands. Now these bands have motile cilia, and they keep beating very strongly. So you can see a high-speed video of the band, and you can highlight that the bands are actually scattered all over the body. When you focus on one of the arms of the larva, right, we actually see that cilia beat in one direction here, and in this region, the cilia beat in another direction. So there is a net flow upward, but when you zoom in, there are these reversal regions that we see. Now we saw this for the first time. Okay. If you zoom in further on these reversal regions, you see that there are these two possibilities. We call them tangle and display. They're not defective, but it's just like physics defect terminology. Okay, so there's this reversal region. So when cilia beat away from each other, you have a net inflow into the surface. When cilia beat toward each other, you have a net outflow from the surface. So now the beauty is, if these defects are all around the swimmer and you can capture the boundary nicely, we can actually use this in a mathematical model, right? So what we did was go to, and of course, I kind of forgot to mention, these defects are the first fundamental reason why these vortices form in the first place, okay? They are the source of all these vortices. So now, as I said, when you have these conditions, like the sign changes on the boundary, you can actually use a square model. So this is a two-dimensional square model cross-section of a cylinder. And this was developed by John Blake in the 1970s. And he analytically derived expression for velocity as a function of the boundary Fourier coefficients. Okay. So now in literature, people always used the first mode, the B1 mode, which is the swimming mode, which gives us two vortices. And we always neglected higher order terms because it is not relevant, right? But now we found that if we go to the fourth, fifth modes, you can actually recapture the swimming flow field. So imagine such a Starfish system, we have an analytical mathematical model to capture the flow field. And that's very, very powerful, right? So that's what we did, which was really nice. And so this was now really nice. We had a model for how the vortices form. And now let's look at why, right? We want to see biologically why this is relevant. So it turns out these animals, this larva, feed using this strategy. So all these wiggly trajectories are algae, this white wiggly trajectories. And you can see when these wiggly guys come near the vortices, they kind of get trapped, right? So they get trapped in the vortices. So this swimming algae are swept up in the vortices, and they are eventually brought to the mouth of the larva for feeding. So the importance of having these vortices is for feeding, actually, okay? Now with this simulation, so we have this model, so we can make simulations like this. So we can vary the number of vortices, right? 0, 4, 6. And we have a line source of food particles incident from the right. And we can show that if you have more number of vortices, there are more repeated chances for the particles to stick on the surface. So higher number of vortices increases feeding efficiency. You can show that from the model. And there's a compromise. The compromise is that the swimming efficiency actually goes down. So there is a trade-off between swimming and feeding. And this is really an interesting thing that we discovered. And it kind of makes sense in the real world because these larvae are in the ocean, right? So if they come across a nutrient patch, rich nutrient patch, they turn on the feeding mode so they can eat a lot. But the moment this patch goes away, they have to go look for the next patch, right? So then they put on the swimming mode. So this is the summary that ciliary bands generate complex arrays of vortices, and these transient ciliary defects underlie vortex formation. And the idea is that hydrodynamics actually determines feeding swimming trade-offs. Okay. And once again, connecting the form versus function theme, the larval morphology gave rise to these vortices, and these are responsible for the functional feeding versus swimming behaviors. And we saw when we zoomed in that the vortices start at the smallest scales, and they result in this emergent large-scale phenomena. So in my lab here, yes, we're really interested in, so there's so much of diversity in larval forms and ciliary bands. So we're really interested in pursuing so many different questions on these exotic marine critters. So what we studied was this starfish larval system, but look at the diversity. It's amazing. It's an amazing ocean out there. and we want to study fluid mechanics in different systems. So that's one thing we're pursuing in our lab. So here is an example of a collaboration. So I can stop in like 4 or 5 minutes, definitely. So this is a recent collaboration with the lab at UT Austin, John Wallingford's lab. So they work on frog development, and they looked at the tadpole stage. So here, if you take one of these frog tadpoles, the skin is what we're showing here. This green, it has these green patches, and those patches are actually cells with multiple cilia. So these are multiciliated cells, and these cilia are motile. Okay, so if you go near the surface of the embryo, let's say you go here, and you put some tracer beads on a microscope, you can actually image the motility of the cilia, just like we saw in the starfish. So you can see there's a nice ciliary flow field. So Mia was my classmate in MBL. I did this embryology course with different developmental systems. So she already knew that I'd be interested in putting in beads and everything. So she did that. And then the beautiful thing now is this is the wild type embryo. So she carried out a specific perturbation for a motor protein called KIF9. So she developed a morpholino, she perturbed it. That means she disrupted the ciliary function. So when she does a disruption, you see there is no flow, but in a wild type embryo near the surface, there is a strong directional flow, right? She also did the rescue, and in the rescue, they actually can recover some of the function. So what we did, my student, actually Bikram did, comes my first grad student, is quantify these 3 things. So here you can see qualitatively that the flows are different, right? But we did this more quantitatively using PIV near the surface, and we could actually extract numbers and show that wild type versus knockdown. Knockdown is really effective. It really perturbs ciliary function, and you can also rescue the function. This is one of our first papers from our lab in terms of a collaboration paper. It just got accepted 2 days ago in Journal of Cell Science. We're also working on fluid mechanics problems. So during the pandemic, we spent so much time in the kitchen. So we wrote, my friends and I, Arnold was also fellow postdoc at Stanford. Now he has a lab at UPenn. Maciek is from Poland, and Endre is at Sweden, at Norway actually. So we wrote a review paper on culinary fluid mechanics. So there's so much fluid mechanics in the kitchen, and there is actually no one source, single source of review. So we wrote about everything, starting from when you open the kitchen tap to what phenomena you see in the sink, to making dishes and let's say cocktails like this is champagne, this is a cocktail. You see Rayleigh-Taylor instability and all kinds of interesting things. So you can go check out this review on arXiv. It's a very comprehensive review paper. I'm very collaborative, as you can see. So I have collaborations all across campus. I collaborate with biologists, Professor Brown and Atula Vikramanayake. At Erasmus, where I hold the other secondary appointment, I collaborate with Professors Diego Lehrman, Langdon, and Baker. Also with Brian Horse and Claire Paris. In engineering, I collaborate with Ashu Agarwal, and Pranay and Landolf. I'm also starting this collaboration in the School of Medicine with Dr. Priya Rai. Show you 2 slides on what we're kind of working on. So this is Patrick Kale. He's diving. He's a 3rd grad student, and he's now working on these local Acropora coral species. This is native to Florida. And this collaboration is with Pranay and Landolf, Ian Enox at NOAA, and Diego Lehrman. We received this recent Euling award to answer the question, how and why is coral growth affected by temperature variations and pH variations? And what we're doing is taking this engineering microstructure approach and also looking at local fluid mechanics. And the idea is to kind of overall have more resilient corals and to understand how temperature and pH affects them. So this is one line of research. The other research we're starting is to look at how we can develop cancer cures using an engineering approach. This is again with Pranay and Landolf and with Dr. Priya Rai at Medicine, Medicine. So here we want to look at how we characterize the biophysics of single cells, look at control healthy cells versus oncogenic perturbations in cancer cells, characterize really the morphology, motility with live imaging, characterize mechanical properties, and Landolf is an expert with tensegrity analysis. We want to use those types of models to build predictive mathematical models for both diagnosis and treatment of cancers, especially when you have few cells, such as circulating tumor cells. So this is an exciting project we're starting. So yeah, I guess acknowledging spoke a lot about my postdoc work, so acknowledging my postdoc advisor and the 2 grad students I work with. And our group is growing in size. The 4th grad student is coming in fall, Gopika. And in a few years' time, I hope I'll give you updates on what Bikram and everybody is working on. We have a postdoc coming in also, computational postdoc, and plenty of undergrads from, again, all kinds of disciplines, engineering, Erasmus, and biology. So I guess I'd like to thank the lab and my postdoc lab. And yeah, that's it. So thank you. If you're interested in anything with marine systems and biophysics, always please feel free to reach out, and I'll take any questions at this point. Thanks. Thank you.